Hello everyone. Today we are going to talk about GI tract pathophysiology. So before diving into the clinical part, let me recap what we have learned before. So first we learn about the physiological functions of saliva before diving into the pathology part. So saliva is not merely water, it is a complex exocrine secretion. So remember that's exocrine secretion critical for digestion, lubrication, protection, and maintain the chemical balance. So lubrication and bolus formation. So it coats the food particles and mucous surfaces to minimize the friction. So it is rich in mucins. What are mucins? High molecular weight glycoproteins secreted by submandibular and sublingual glands. So mucins bind water, providing viscosity and elasticity, which facilitates chewing, bolus formation, and smooth swallowing. So, and also saliva provides enzymatic digestion. So, contains alpha amylase, that means taurine, secreted by parotid glands, which cleaves alpha 1 4 glycosidic bonds in starch, and lingual lipase created by Ebner's gland, which initiates lipid hydrolysis in acidic stomach environment. So remember this will initiate lipid hydrolysis in acidic stomach environment. Though this lipase, lingual lipase, is secreted by Ebner's glands in the mouth. And then, so this is in saliva. This lingual lipase and alpha amylase. What is this alpha amylase? It is taurine, secreted by parotid glands. And antimicrobial and immunological defense. So it inhibits bacterial, viral, and fungal proliferation in the oral cavity. So it contains secretory IgA immunoglobulin that prevents pathogen adherence, and also it contains lysozyme that clears the bacterial peptide gland cell walls, and also it has lactoferrin that sequesters free iron needed for the bacterial growth, and histidines. So it is a potent antifungal protein targeting Candida albicans. And it also has a pH buffering capacity. That means it neutralizes tartaric acids and prevent tooth enamel demineralization. So this is really important. So this is mainly driven by the bicarbonate ions whose concentration increases dynamically with salivary fluoride, also aided by phosphate buffers and saliva. What is this? A basic peptide that raises the pH by producing ammonia. And also tooth remineralization and integrity. So constantly it repairs microscopic dental lesions and hardens the enamel. So saliva is super saturated with calcium and phosphate ions, stabilized by proline-rich proteins and satharine. This maintains a chemical gradient that favors the remineralization of hydroxyapatite crystals. So remember, these are the functions of saliva. So remember, there's a syndrome called Sjögren's syndrome that often targets the lacrimal glands. So that means it causes dry eyes, dry mouth. And also, you have to check for the salivary gland swelling to check this disease. And next, we'll go to Xerostomia and its clinical implications. So, This xerostomia is the subjective sensation of the dry mouth, while often caused by quantitative reduction in the salivary flow rate. That means hyposalivation. It represents a profound loss of the local physiological protection. So, what is this xerostomia? So, this is basically dry mouth feeling with multifactorial etiologies. So, this can be due to autonomic dysfunction, the salivary actinal cell destruction, example, the Sachron's syndrome, and head and neck radiation, or pharmacological effects, side effects like anticholinergics blocking the muscarinic M3 receptors on the sinus walls so that it prevents fluid secretion. So these are the causes for this. And also dental caries and enamel demineralization. So accelerated and aggressive decay of tooth structures. So absence of salivary bicarbonate prevents neutralization of the lactic acid produced by Streptococcus mutans. So when the oral pH falls below 5.5, hydroxypeptide dissolves. Hydroxyapatite dissolves. So lack of salivary calcium and phosphate prevents remineralization. And then severe dysphagia. So difficulty swallowing food and clear articulation of speech. So without water and mucus, the coefficient of friction on the tongue, cheeks, and pharynx increases exponentially. So food cannot be formed into a coherent, shapely bolus. Friction-induced pain inhibits the voluntary phases of swallowing and normal lip or tongue speech mechanics. And then opportunistic infections, oral candidiasis. The frequent development of trash uh, mucosal or soreness. So loss of histidines, salivary IgA, and lactoferrin removes natural biological breaks and fungal overgrowth. So Candida albicans. So normally a benign commensal colonizes the mucosal surface, invading the epithelial cells and forming white pseudomembranes. And also altered taste. Dysgeusia. So severe reduction of metallic dissolution in taste perception. So that means taste buds are located in the deep crypts of papillae and require taste tends to be fully dissolved in a fluid solvent to reach the sensory receptors. Additionally, saliva contains carbonic anhydride, VI, that means 6, or gustin, a tropic factor essential for taste bud growth. Loss of it also lead to taste bud atrophy. And ulcers formation, that means painful cracking of oral tissues and chronic fall breath. So salivary epithelial growth factor, that means EGF, when it is absent. So it delays mucosal cell turnover and wound healing. So without salivatory washing action, dead epithelial cells accumulate. Anaerobic bacteria ferment these cells, releasing volatile sulfur compounds like hydrogen sulfide. And then we're going to talk about the cephalic phase of this. So gastric secretion, it occurs in three phases: cephalic, gastric, and intestinal. So side of food activates the cephalic phase, repairing the stomach biochemically active food digestion before even food enters. So how does this happen? So first it happens via sensory activation and neural pathways. That means cognitive, visual, olfactory, or gustatory stimuli trigger this gastric priming. That means sensory inputs are processed in the cerebral cortex and the appetite centers of the hypothalamus and amygdala. These higher brain centers send excitatory signals to the dorsal motor nucleus of the vagus nerve in the middle. So the dorsal motor nucleus of the vagus nerve, that means cranial nerve 10 in middle. These higher brain these send the sensory information are sent to the dorsal motor nucleus of vagus nerve. That means cranial nerve 10. These are sent by higher brain centers like hypothalamus and amygdala. And also vagal efferent outflow. So parasympathetic vagal stimulation triggers multicellular gastric response. So vagal fibers, they travel to the gastric wall where the postganglionic parasympathetic fibers, postganglionic means after the synapse, and parasympathetic fibers release acetylcholine. So this acetylcholine targets the muscarinic or M3 receptors on the parietal cells. Acetylcholine targets M3 receptors on the parietal cells. So which nerve does this? How are they acetylcholine is produced? Because the vagal fibers travel to the gastric wall to the place where the postganglionic parasympathetic fibers release acetylcholine. So this acetylcholine then targets the muscarinic M3 receptors on the parietal cells. So that's the direct pathway. And there's an indirect pathway as well. That is enterochromaffin-like cells, ECL. So that is an indirect pathway. So then there's this ECL cell activation, that means histamine. So histamine acts as the most potent paracrine synergist of acid secretion. That means this vagal acetylcholine binds to the M3 receptors on the ECL cells or enterochromaffin-like cells, stimulating them to secrete histamine. So these ECL cells will secrete histamine. Histamine diffuses locally to bind to H2 receptors on the nearby parietal cells, activating adenylate cyclase, raising intracellular CAMP and driving H plus or K plus ATPase proton pump. So this activates adenylate cyclase, and then adenylate cyclase do what? It will make CAMP and then protein kinase and so on, activating proton potassium pathway or ATPase pump ultimately. So now you should remember how this happens. So histamine diffuses locally to bind to the H2 receptors, and then it will activate due to adenylate cyclase, that means CTP activation, and that will produce CAMP and thus the protein kinases. Drive the H plus and K plus ATPase proton pump. And then when we talk about the G cell activation, gastrin. So vagal postganglionic terminals in the antrum release gastrin releasing peptide. You must have heard of this as GRP onto the G cells. So G cells then release gastrin into the blood. Gastrin binds to CCK2 receptors on both parietal cells, that means direct pathway, and ECL, enterochromaffin-like cells, indirect. Why? It causes histamine release that will ultimately stimulate T cells. And also, but another thing that it does is D cell inhibition. That means somatostatin. So this is like lifting the natural biological brake on acid secretion. So vagal acetylcholine binds to the muscarinic receptors on the D cells, inhibiting the secretion of the somatostatin. So it will inhibit the secretion of the somatostatin. Under the resting conditions, somatostatin tonically inhibits T cells and plasma cells. So vagal activation suppresses this inhibition, allowing maximal acid out. So somatostatin is like an enemy. It doesn't allow this to happen in resting conditions, because in resting condition it inhibits T cells and plasma cells. But after vagal activation, it suppresses this inhibition, allowing maximal acid out, because it will inhibit vagal acetylcholine will inhibit somatostatin. Somatostatin is where in antral D cells. So when somatostatin is inhibited, it can't inhibit T cells. So T cells now can produce gastrin. Hope you understood. So first acetylcholine binds acetylcholine goes to the antral D cells. D cells are the ones that secrete somatostatin. If somatostatin is there, it will go and inhibit T cells and plasma cells. So as now vagal activation, that means acetylcholine go and bind to this ACH, it won't be able to further stimulate this activity.